Welcome to the series I'm simply going to be calling Sciencing, intended to help you to better understand scientific research. This module introduces you to a type of experimental study design known as the randomized controlled trial. This is the gold standard for assessing if a given intervention, a given treatment of some sort, for example, diet, exercise, whatever it might be, has a specific effect or set of effects. It is intended to test hypotheses and essentially confirm or not a causal relationship between the intervention, which is now we're talking about experimental studies, replacing the idea of exposure. We're determining the exposure, we're calling it an intervention, between the intervention and the outcomes of interest. In terms of primary research, so original work, the randomized controlled trial is at the top of the research evidence hierarchy. Or rather, at the very top, there is a synthesis, preferably mathematical synthesis of multiple randomized controlled trials. But basically, as I say, in terms of primary research, not reviews of existing research, but original work, the randomized controlled trial sits at the top of this hierarchy. This is, however, only correct when it comes to testing interventions, as this is what randomized controlled trials are designed to do. If we want to know the prevalence of something, a cross-sectional study design is more appropriate. And if we want to know the incidence, a longitudinal cohort study design is more appropriate. But for finding out what treatment is called for in a given situation, we're dealing with, or should be, the randomized controlled trial. The types of questions that randomized controlled trials are suited to answering include how well does this intervention or this treatment work relative to some other option with respect to a given outcome? And also, what are the side effects of A relative to B, whatever A intervention and B intervention might be? In other words, they tend to focus on assessing the efficacy or effectiveness, and or they assess safety, sometimes also called harms, or what you might think of in terms of side effects or adverse effects. In theory, they should do both, but in reality, safety-related results are often poorly reported, unfortunately, unless this was one of the key goals of the study. And also, rarer adverse effects or rarer side effects may not show up unless the study has a very large number of participants. The 1 in 1,000 events, or 1 in 10,000 events, or 1 in 100,000 type events will only likely show up if there is a large enough group. And given we will want to compare between groups, it has to be a meaningfully large number to allow some kind of statistical analysis. If we do have 10,000 in one group, 10,000 in another group, and one person in one group had an adverse event, it's very hard to necessarily attribute that to the treatment because it's so rare. The specific questions in a study might be worded a bit differently, use different wording to what I've used here, but they always assess the effect of one intervention or treatment versus another, even if the other intervention is a placebo or the status quo. Although the majority of randomized controlled trials use only 2 groups, they can use more. For example, 2 different interventions might be compared to an inactive control. For example, aerobic exercise versus resistance training versus no exercise. Now, although there is no theoretical limit to how many interventions could be compared, in practice, it's rare to see more than 3 or 4 in the same study, and the most common number is 2. In fact, the minimum number is 2, or we're not dealing with a randomized controlled trial. The essential elements of a randomized controlled trial are in the name. The participants are allocated to the intervention or treatment at random. And there are 1 or more control or comparison groups. Other common but not universal aspects such as blinding will be discussed in another module. We'll address these two core characteristics in reverse order. Both randomization of study participants to interventions and having control or other comparison groups are intended to isolate the effect of the intervention from anything else that might explain the study findings. So, classic scientific method. Having a control group accounts for things related to time, for example. Without some form of comparison group, all we can do is measure the difference, if any, before and after our intervention. And that presents some serious problems. Let's say you want to know if taking 1 gram of vitamin C every day for the duration of a flu infection, so influenza infection, helps people to recover more quickly. Well, we know that most people will recover with or without the vitamin C. So just having one group doesn't help us. We need to compare those taking the vitamin C with those not taking vitamin C and see if there is a difference in recovery times between the two groups. Or we might want to know how well a quit smoking support program works. Here, the problem might be that while our intervention is specific to our study participants, other public health campaigns on television and radio and in pamphlets at GP practices may also be influencing our participants. So without a control group, we wouldn't know if our intervention was effective or one or more of the other campaigns our participants have been exposed to should get the credit for any change. By having a suitable control group or comparison group who do not get our intervention but are also just as likely to be exposed to the public campaigns, we can assess whether our intervention was likely to be the reason for any difference. So, why randomize? We assign study participants randomly in the hopes that each group has essentially the same spread of people, meaning we want the average of each group to be similar so that any differences we observe between groups at the end of a study is likely to be due to the intervention rather than some fundamental differences in the groups being compared. Using our vitamin C example, if instead of randomizing people, we put mostly young, generally healthy people in one group and mostly older, less healthy people in another group, and we gave vitamin C to the younger, healthier group, if we observed a faster average recovery from flu in the vitamin C group, we wouldn't know if it was because of vitamin C or because younger, healthier people recover faster. Or if we found that vitamin C group had a slower average recovery time, we couldn't be confident that the vitamin C was harmful, as it may be that the younger, healthy people only get sick from worse flu strains. Why not allocate so that all groups are similar? It seems reasonable to work out the factors we think may influence the outcome other than the intervention and then intentionally match participants. In our vitamin C example, we could ensure equal numbers of young, generally healthy people and older, not so healthy people in each group. The problem is that there may be other important factors that we don't even know or don't know about that also influence recovery from the flu that our process doesn't address. Randomization is about trying to ensure both known and unknown factors are accounted for. Basically, we want to ensure that as much as feasible, we isolate the effect of our intervention from confounders, meaning other things that may influence or do influence our outcome of interest, other factors other than the intervention or treatment. It's impossible to do this perfectly, and that's why we shouldn't get too excited by the findings of 1 or 2 randomized control trials, especially if these studies maybe only had a few dozen participants. And we should not use study proves or studies prove. Studies always suggest. Science is a game of probabilities and confidence. Yes, we can grow in confidence as more and more studies come in with basically the same results. But there could be some fundamental flaw in those studies. So our confidence grows, but if we're becoming absolutely certain we're moving away from science and into something else. So given the importance of participant randomization and appropriate comparison, key questions when assessing randomized controlled trials therefore include: how is randomization achieved, and how random is that method really? Some things are not as random as they seem. Can we have reasonable confidence that the groups are similar? So they should really be assessed after randomization to see if at least those factors we think might be important, other than our intervention, are reasonably evenly distributed. In theory, randomization will take care of the rest, but we should at least assess if it worked for those things we think are likely to be an issue. And finally, were the control or comparison groups, or the comparison group, appropriate given the aim of the research? What are we comparing against? If the standard of care in a country is a given treatment, and someone wants to find out if something else can be effective, rather than working out if that new thing, that new intervention is better than nothing or a placebo, it would generally make sense to compare it to what is already being done so that we can find out, is it as good? Is it better? Is it not as good? And potentially, is it not even effective? So again, it depends a lot on the question being asked. There are a number of types of randomized controlled trial designs, or sub-designs if you like. The ones we'll look at are parallel, crossover, and cluster. The most common approach is the parallel design. Here, participants are divided into 2 or more groups. Each group is given the intervention or the treatment specific to that group until the study finishes. The outcomes of interest are assessed at the beginning and at the end, and sometimes they're also measured at time points in between. Sometimes a crossover design is feasible. Here, we still have random allocation of participants to a group, but at some point the intervention is flipped so that the group that got intervention A now gets intervention B and vice versa. Usually with a period of time in between. This is the ideal, as each participant acts as their own control or their own comparison. So we're getting rid of the problem that in a parallel design, I'm different, you're different to everyone else, at least a little bit. But here we can compare me to me and you to you under different conditions. Unfortunately, this is not always practical or ethical in all situations. So we need to consider feasibility before we say, oh, this was a parallel design and it should have been a crossover. But it may work for a treatment that manages, or treatments that manage a condition, for example. It isn't always appropriate to divide individuals into different groups. For example, if we wanted to know how well a new system for monitoring babies on a neonatal intensive care unit worked, we would be better off comparing hospitals. In this scenario, each ward or hospital is a cluster, hence the name cluster randomized control design, or cluster design. Other than that, the design works much the same as the clusters are randomly allocated to each intervention. We'll end briefly on analysis. When looking at the results of a randomized controlled trial, we should focus first and foremost on the comparison between groups, not within groups. Within groups refers to before and after, but between groups refers to often change in group, before and after, versus change in another group, before and after. We should be very suspicious when a research article describing a randomized controlled trial emphasizes within group results. Nothing wrong with reporting them, in fact they should be reported, but it shouldn't be the emphasis, because that defeats the purpose of having a comparison group in the first place. The whole point is to compare between groups. If you found this useful, If you found it interesting, please hit like, hit subscribe, and most importantly, keep watching other modules to keep learning about this really important topic that affects really all of our lives. And some examples of that are here.